Okay, it's 7 o'clock. We could get started. Hi, I'm Musella, and this is our Brain Tumor Awareness Month webinar series. Tonight's special guest is Dr. Harry Gruber, MD, from Tocagen. He's the president. And tonight's topic will be Toca 511. Dr. Gruber, take it away.
Thank you, Al, and thank you for the Musella Foundation for setting up this webinar series. We're going to be talking about Tocagen and our preclinical as well as clinical data. Tocagen stands for 2 cancer genes. That's because our core platform is to develop products that deliver genes directly into cancer cells. We'll be starting with a discussion of the scientific rationale of our proposed mechanism of action based on our preclinical data. Then we'll talk about the phase 1 clinical data and then turn to what further clinical development of Toca 511 and Toca FC we are planning. So to start with, we use a retrovirus. It's a replicating retrovirus. It's abbreviated RRV, and it functions as a gene therapy platform. And the reason it's we call it cancer selective is because it efficiently infects cancer cells. And you can see in the bottom left panel here, the brown stained cells are the staining of the virus proteins. And you can see the cancer growing, brain cancer growing in a mouse brain is stained brown, but the contiguous normal brain is not stained. So the virus is truly highly cancer selective. And the reason it's selective is because the cancer is bathed in white cells that make interferon. And so the cells that grow out are interferon pathway resistant. And our virus is cleared by the interferon pathway. So in defective cells, like cancer cells, our virus can grow. But in normal healthy cells, the pathway of interferon clearance is intact, and this virus is not a known human pathogen and does not carry offense to knock out the interferon pathway, so it only grows or selectively grows in the cancer cells. Another feature of this type of virus is that it infects the cell, integrates into the DNA, and then gently butts off infectious viral particles, which then can migrate to other cancer cells and infect them. So at a high level view, at a patient level, the virus is administered to a patient either at the time of surgery into the surgical bed, or it can be given transcranial into the bed, or intravenously. And yes, it does go through the bloodstream and get through the blood-brain barrier into brain cancer tissue. Once the virus is given, it not only infects the cancer cell and spreads through the tumor, but it generates a gene product called CD, or cytosine deaminase, which we'll come to. After 6 weeks of viral spread, the patient then takes oral tablets called Toca FC, and the Toca FC is made by the CD protein in the cancer cells into 5-FU. The 5-FU is a very well-known anti-cancer drug, and it kills cancer cells. And with the killing of the cancer cells, and there's releases of tumor-associated antigens, and in combination, as the cancer cell dies, there's a release of viral proteins or pathogen activating molecules, sometimes thought of as adjuvants, and there are also PAMPs. There are also DAMPs that are released, things that are you would think of as toll-like receptors. And all 3 of these elements then combine to prime and activate the immune system against the cancer. Over time, the 5-FU also kills these red cells here that are called myeloid-derived suppressor cells. As these cells are killed, the immune system dramatically activates, increasing the number of CD4 and CD8 T cells as the myeloid suppressor cells go away. At this point, I wanted to share with you a brief animation so you can get a sense of how this is playing out over time.
Tocagen is developing broadly applicable cancer-selective immunotherapy treatments for cancer, including high-grade glioma, a type of brain cancer. The company's lead product combines Toca 511 with Toca FC. The therapeutic regimen involves 2 discrete steps, with Toca 511 given first, followed by treatment with Toca FC. During the first step, the patient receives Toca 511 by injection. Toca 511 is a replicating virus that infects actively dividing cells. In normal adult tissue, where most cells are not actively dividing, Toca 511 replication is further inhibited by the immune system. In contrast, cancer cells are actively dividing and having defective immune responses, providing a safe haven for Toca 511. Toca 511 delivers a gene for an enzyme called cytosine deaminase, or CD for short, to cancer cells. As it spreads through the tumor by budding and cell-to-cell contact, Toca 511 programs cancer cells to make CD. These cancer cells are now primed for the Toca FC step. Patients then take a pill called Toca FC. Within infected cancer cells, CD converts Toca FC into a potent, FDA-approved anti-cancer drug called 5-FU, which kills cancer cells. 5-FU also kills MDSCs, which are cells that help tumors avoid the immune system. The immune system is further activated by tumor-associated antigens and viral proteins released from dying cancer cells. As a result, immune cells are activated selectively against cancer cells. This approach selectively destroys cancer cells within the body, while leaving healthy cells unharmed. Patients take multiple cycles of Toca FC, repeating the sequence of events, which may ultimately result in tumor destruction. Tocagen believes no one should die of cancer. Visit tocagen.com to learn more.
So with that, I think everyone's an expert now in the mechanism of action of our combination product, and going to go a little deeper for questions that are commonly asked of us. So the yeast cytosine deaminase gene is inserted into this viral sequence. As you can see, the yeast protein, there is no equivalent of that in humans. So the cancer cells are the only cells in the body that have the ability to convert the 5-FC, as we've talked about, into 5-FU, which is the anti-cancer drug. In terms of the selectivity of the virus for the tumor, that leads to selectivity of the location of the 5-FU. And you can see here the rat glioma model, when treated with our combination treatment, generates very high levels of 5-FU in the tumor. This is approximately 100 times the IC50 of what it takes to kill a cancer cell. And yet in the blood at the same time, there's essentially almost no detectable 5-FU. In contrast, when you look at humans who receive intravenous 5-FU the way it's normally given, you only get about 100th the amount of 5-FU in the tumor, and really barely enough to do much value. And at the same time, there's huge amounts of 5-FU in the blood, which leads to killing of dividing healthy cells, such as gut lining cells and bone marrow cells. However, with our treatment, we've changed the biodistribution from the blood into the tumor. And so we don't expect, and so far do not see, any of the side effects you would normally see from 5-FU. The reason that there's so little in the blood, that as soon as the 5-FU leaks out of the tumor, it's rapidly metabolized in minutes and is therefore eliminated outside the tumor, but it's continuously made and available inside the tumor. The time course that we've been talking about is can be seen here in this experiment, data from experiments where the 5-FC is given to tumors, to animals with tumors that have the 511 already spread through them. And then we sample at days 3, 6, 9, and 14. And you can see at day 3, not much has happened. At day 6, there's a dramatic elimination of the myeloid-derived suppressor cells and other myeloid suppressor cells called TAMs and monocytes. And only after this point that the break's removed do we then see a gradual reduction to complete elimination of the tumor. And at the same point in time, we see the elevations in CD4 and CD8 cells, as well as B cells, all immune cells that are involved in tumor destruction. The suppressor cells, the T suppressor cells, are not affected by this treatment, which is good. And we've seen all of these effects in various models, including intracranial glioma models, subcutaneous models, and liver metastatic models. In terms of the glioma, when we initially looked at this treatment in mice that have no immune system, nude mice, with a human brain tumor growing in them, the tumor normally would kill these mice in about a month. If the animals, though, these same type of mice are treated with the virus, allow it to spread, and then with the FC, the tumor is almost immediate eliminated. And you can see the tumors carry the firefly gene, and you can see the light emitted as soon as the tumor is killed. You don't see the emission of light. However, there are always a few cancer cells left behind. They're often thought of as cancer stem cells that are non-dividing, and 5FU only kills dividing cells. So these few cells can essentially recreate the entire tumor in about a month. But if the mouse is continuously treated approximately every month with the 5FC, you can control the tumor continuously, and the animal lives near a normal life. Now, in contrast, in an animal that has a normal immune system and a murine cancer, brain cancer cell line, the tumor is established, the virus is administered, the 511, and then the tablets are given. Now, in contrast to the mouse without immune system, those with an immune system, after the 5FC treatment, after a couple of cycles, you can stop the FC and the mouse lives a normal life. And so we interpreted this as most likely related to activation of the immune system against the tumor, which we subsequently confirmed in a variety of experiments. And so, for instance, you can re-challenge the mouse with the same exact tumor in their flank, and without other treatment, this red line, which is the volume of the tumor clears gradually over time and does not come back. The black line represents animals that never were cured of their tumor, and then the tumor obviously just grows without treatment until the animal is sacrificed. Another example of this type of immune activities, we can take mice that have been cured of their tumor and take lymphocytes from these mice and infuse them into mice that have brain cancer that would otherwise be lethal, as you see in the blue line. However, if they get the lymphocytes in green, in this experiment, about 70% of the animals had eradication and other tumor and long life. So it shows that it is the white cells that are responsible in these models for the beneficial effects. In addition, through experiments to eliminate different subgroups of white cells, we've been able to show that the effect is almost completely generated by the CD4 helper cells, which drive the identification of antigens for naive white cells and help drive a immune response broadly based against the tumor. Now we're going to turn to the human clinical data. And I'm sure this audience is very familiar with the course of brain cancer. In our initial studies, we started in those patients who had been diagnosed with brain cancer, received standard of care, which includes surgery, radiation, and temozolomide. And then unfortunately, the tumor recurred at about 8 months on average, and they entered our study, and they received our treatment. It was an ascending dose safety study, but of course, we were also looking for drug activity. And on average in humans, from recurrence to death is typically 8 months. So in these 3 studies that we did, we looked at 3 different routes of administration of the virus. One was at the time of resection. We injected the virus into the tumor that was left behind at the time of surgery. In other patients, we did intratumoral, which is direct transcranial injection of the virus into the tumor. And finally, in some patients, we gave the virus intravenously through the bloodstream. And as I said earlier, it does populate the cancer cells in the brain tumor and therefore gets through the blood-brain barrier. The safety profile of this combined treatment has been very promising. Among these 3 studies, we've evaluated 127 patients, and you can see in the grade 3-4 category, we have extremely low percentage of treatment-related adverse events. In terms of the survival, the virus, of course, mechanistically should be working about the same. The virus is there, and then they get the FC. And so in all 3 studies, we saw approximately the same median survival, which is around 13 to 14 months. We're going to spend the rest of our time in this phase 1 study on the resection study, because these patients were the ones who were most interested initially into getting into the study. They wanted their tumor out, they wanted to get treated, and this group did seem to survive a little bit better. Of course, this is not a randomized trial, so we can't directly compare them. So in this patient group of the resection and injection at the time of resection, you can see these patients were mostly the more advanced type of tumor, the GBMs, as opposed to the 11% with anaplastic astrocytomas. Most of the patients were first or second recurrence, but about a quarter to a third of patients had 3 or greater recurrences. And so some of these patients were quite advanced. This paper data was so compelling that it was, even though it was a phase 1 study, it was published in Science Translational Medicine, and you can tell from the animation on the cover that they used our work for their cover display. To highlight some of the factors that we reported in this study, obviously the favorable safety profile I already explained. There was increased survival versus historical benchmarks. There were 6 durable CRs that all remained alive at the time of publications. And within the phase 1 patients in the resection study, 5 of 23 had complete responses, meaning by independent radiology review, all of the tumor, all the brain cancer tissue went away and stayed away, making them actually durable complete responses. And that's 22% of the patients. There were also patients with stable disease. So the overall benefit rate, including CRs and stable disease, is over 43%. The median survival for this group of patients is 14.4 months. This is the group with the entry criteria identical to our subsequent phase 2-3 studies. And overall, at 3 years, there was over a quarter of the patients were alive. So, as I said earlier, if you look on the left-hand side, you can see various different treatments and the median overall survival when used in the recurrent setting, and it ranged anywhere from about 6 to 9 months. We did use a dose in the human studies that was effective in mice, but only about half effective, and that's these lower 3 cohorts. And so you can see some apparent benefit with these patients living a median overall survival of about 11.8 months. And then at the higher dose, as I said, this is where we saw the 14.4 months. So we saw a dose response on median survival. There's no external control, but internally you can see there is a difference by dose. In terms of the response rate, the study was initially planned for 6 months observation period. Once that we noticed that patients were living a lot longer than the 6 months, we were able to continue to follow them. And so in the ascending dose study portion, we saw 6 patients out of 53 that had responses, and in just those that had the higher dose, there were 23 patients. This was also restricted. This group also were the ones who had first or second recurrence and a 5 centimeter or less tumor size, which was most of the patients in this high dose. And you can see that there's the 22% CRs and the 43% clinical benefit rate. Of very high importance, typically in this type of setting, you see 1% or less CR. So this 22% is very different than what the reported literature is. In addition, these patients with CRs lived a median so far up till this reporting of over 3 years. Other drugs that are used in this method, bevacizumab, Avastin that is, or lomustine, we're in the 4 to 6 months. So what we're seeing here is something much more durable than what is typically seen. So as we followed these patients, all of them in the 23 group, you can see their survival is quite long. Here at 8.4 months is the average of 4 major lomustine studies. And you can see most of the patients in this study, this portion of the study, live more than the 8.4 median survival. And in fact, you can see just how long these patients who had CRs, and even one patient who didn't achieve a CR, they're out in the 3 to 5 year time frame. These are months of survival, not the normal weeks that you would see in these types of studies. And we saw a benefit by this criteria in both patients with GBM as well as those with grade 3 or anaplastic astrocytomas. And what was really interesting is that these tumor shrinkages began at around 6 months, but complete response in some patients, like this patient here, number 18, didn't have a complete response until about 30 months out. And the stars are the patients who are still alive at the time of this reporting. And I think it's important to understand what we're really talking about in a surgical setting about responses, because it is helpful to see these MRIs. So here's a patient on the far left who, in white, is his tumor that came back about 5 months after his resection. He took about a month or so to get into our trial, and you can see the tumor is growing. So this is an active tumor at this point. At the time, right after the surgery, you can see most of the tumor is taken out, but there is a region left behind in the speech center. They didn't want to interfere with his ability to speech, and they were hoping this treatment would provide benefit to clean up the tumor left behind. And so the patient goes home after the resection, and the tumor is being infected with the virus as the virus spreads during this 6-week period till we then get a baseline MRI. And you can see even during this period, the tumor appears to be growing because there's no 5-FC that's given so far. And then the patient comes back 6 weeks later, and you can see the tumor is stable, maybe a little smaller, but it was called stable by the independent group. And then every 6 weeks, the patient comes back until finally he has a partial response. And as he's followed longer, he goes into a complete response. And he's out today way past the 3-year mark. One of the things that we did as we ran this study, this had to be done from the very beginning. We designed the study that we, like in mice, we would look at immunological responses. And so one of the things we did is we got a sample of the tumor that was removed before the viral treatment, before the FC, and we found some very interesting signatures, which is that patients had who went on to have responses, meaning complete responses. They had changes in their they had differences in their T cell, their natural killer cells, and their macrophages that were different than those who did not later respond. And you can see here as an example, the CD4 memory cells, activated ones, similar to what we were talking about in the mice studies, were elevated in those who went on to respond. Similarly, there was a reduction in natural killer cells and macrophages in those patients who went on to respond. And so this overall signature, we call it a cancer immune therapy tumor microenvironment signature, had a good correlation with those who responded versus those who did not respond. Now, this study population is small, but it's still an exciting opportunity that we'll be looking for in the registration study that is now running. In addition, we serially received blood samples from these patients at the time, approximately the time of their MRI every 6 weeks. And what we found was in those patients who went on to a complete response, they had an increase that went up and was sustained during the course of their treatment compared to those who do not have a complete response in the what's called E-selectin cytokine. In addition, there was an increase during the treatment course in their MIP-1 beta. Now, this is consistent with an immunological effect in that E-selectin is a cytokine that allows white cells to stick to blood vessels and therefore allow them to more readily get into tumor tissue. In addition, the MIP-1 beta is a chemokine that's released from white cells and attracts CD4 lymphocytes into the tumor. So both of these observations are very consistent with what we've seen in mice. So overall, our combined treatment of Toca 511 and FC activates durable T-cell mediated immune responses preclinically that are CD4 and CD8 dependent, as well as are associated with local myeloid suppressor cell depletion. The 3 phase 1 studies had comparable findings on both safety and survival. And these are both surgical and non-surgical settings. The treatment was well tolerated with very few greater than grade 3 toxicities in looking at the data from 127 patients. In the resection study, there were 6 out of 53 that had durable complete responses. And in the phase 3 eligible population, the 3-year survival rate was 26%, but the longest survival so far at close to 5 years. There were 22% complete responses and a clinical benefit rate of 44%. And we also, I think very excitingly, have an association in both the blood and the tumor that is consistent with an immunotherapeutic mechanism of action. Now, turning to further clinical studies, as I mentioned, we went on to do a study in recurrent high-grade glioma that is a registration quality study. It's scheduled to have its interim analysis this quarter and final analysis in the 4th quarter. We've also been selected by NRG with collaboration with the NCI to run a phase 2-3 registration size and quality study in newly diagnosed GBM. In addition, we are running studies that are still early in a variety of different metastatic cancer types with the same treatment combination. And we have a number of interesting drug candidates based on our RRV platform that we will not have time to discuss today. So in the pivotal phase 3 study, it was designed to enroll 380 patients. It actually, as it's normal, enrolled a little bit extra at 403. And the patients were randomized to either our treatment with the virus followed by the FC, or drugs that are commonly used in this recurrent setting, which is lomustine, temozolomide, or bevacizumab. The eligibility criteria were identical to the eligible group I previously described with GVM or AAs, and they had either first or second recurrence in tumor size at 5 centimeters or less. As part of this study, we are prospectively looking at the lymphocytes populations, immune activator markers on the lymphocytes, cytokines, the number of tumor infiltrating lymphocytes, which we didn't have time to present today, and the immune suppressive myeloid cells. In the newly diagnosed study, the NRG study, again, we're administering the 511 after surgery, but before closure, or the patients receive standard of care, randomized 1-to-1. Those with the 511 get the chemoradiation, which is standard of care, plus the standard of care temozolomide maintenance course, and the standard of care populations, of course, just get the standard of care. So overall, we're extremely excited at the progress we've made to date and what appears to be some dramatic impacts on patients with recurrent brain cancer. And you can see we have two different families that have had children that occurred after the or during the treatment courses, and a family of that has been living very healthy lives, even though they're later in life than these other two families. So with that, I'm going to thank you for your attention. I thank all the families and who help, and the doctors who help run these trials, and the other healthcare professionals, as well as the financial support, and especially Al Musella has been very helpful to us. Al, turn it over to you for questions.
Oh, thank you so much. That was a very interesting presentation. It's a very exciting drug. We don't usually see these durable, complete responses, so that's an exciting thing. So the first question has to do with those long-term survivors. What kind of shape are most of those long-term survivors in? The picture looks like they're young, relatively healthy. Are they able to work?
We don't it wasn't part of the protocol, but they do visit us and they do tell us about that they're working. And even more exciting than that they're working is they're raising families, they buy pets, they go to rock concerts and baseball games. They're living a happy life. And they still have brain cancer, and I'm sure they have some concerns with that, especially, waiting for the MRI every 6 weeks. But we're very excited about just how well they're doing.
That's good. The reason I ask is years ago there was some treatments that had some durable responses, but the patients were in horrible shape. But that's not the case in this place. This is all very good stuff.
Yeah, and not only that, all of the patients who had partial responses went on to complete responses in this study. So that was, I think once the immune system kicks in, it probably has a very important effect for these patients.
Okay. Are there any open studies now for GBM?
With our treatment, no, not at this point. We're working hard to get the newly diagnosed open as soon as possible. And of course, we're interested in evaluating other studies as soon as we have the bandwidth to try to find other settings for these patients, these and other cancer patients.
Is there any opportunity for compassionate use?
We've been asked that question frequently, and of course, we would like to help as many people as possible. At this point, we really just don't have the manpower and ability to provide it compassionate use, but we're looking for the time that we'll be able to make this widely available.
Okay. Have you tried it on pediatric patients yet?
No, it's the same thing as a bandwidth issue with compassionate use or other studies. We've worked closely with designing a study for pediatrics, and we look forward to a time when we'll be able to initiate those studies.
Okay, the concept is so elegant. The question that comes to my mind is, why does it not cure all tumors? What happens?
Yeah, I think that's a very important question, Al. First of all, we learned a lot of things in these first studies. One was that we never saw a response in a patient who had more than 2 recurrences. And we think that over time, the immune system gets weaker and can't be activated, and the tumor gets stronger and is harder to kill. So I think we know pretty confidently we want to treat as early as possible, and that's one of the reasons we're so excited to do the newly diagnosed study quickly. I think, however, there are other reasons that not everyone responds. As I pointed out, the immune signature at the time that a patient is enrolled is different, and so not everyone has the same immune makeup. And there are other factors that could be used by the cancer cell to stop the immune system. Some of them are well described, like checkpoint inhibitors. And it's possible maybe with combinations in the future, we'll be able to see something more than the 22% we're seeing now.
That was going to be the next question, but you answered it, about checkpoint inhibitors.
Yes.
How fast do you see the biomarker response?
This work has been presented in poster form, and so people can look at it. We have this type of data on our site, but in a general sense, we start to see it after the very first cycle of FC. I will point out these are a small number of patients, but it's still exciting.
This is a quick one, easy. What's the definition of partial response?
Yeah, so we use the McDonald criteria, and that's that 50% or more of the tumor has disappeared at the time of the new MRI image compared to the prior.
Okay.
And it also includes that there's no change in steroid dose, meaning no extra steroids, and no deterioration neurologically.
Okay. The next 3 questions are just variations. Are there any chance that the new trials will allow spinal cord tumors? Spinal cord GBM.
Yeah, that's something that is very important. Some of these tumors can invade the spinal cord. It's not something that we're working on right now, but I think given the mechanism of action, it's something we should consider quickly.
Okay, and then will anaplastic astrocytoma be included in that next trial for newly diagnosed?
In the newly diagnosed, they will be enrolled because there's no way to know in advance whether you have an anaplastic or a GBM. Mm-hmm. But the primary endpoint is likely to be based on the GBMs, just because there's so many more of them, and we can't in advance stratify.
And can you guess about when that trial will open?
I'm hoping sometime this year. That's what we've said publicly.
The question is, the virus is only given one time?
Up until recently, the virus has only been given once. There's 2 exceptions to that, but in the phase 1 study now, some of the patients are being allowed retreatment if they ask for them. In addition, in the IV study, we gave the virus IV, then took out the tumor, and as long as we were there, we also treated the tumor bed at the same time.
Okay.
So I think retreatment is something that definitely needs to be considered in future trials.
Okay. Can multifocal GBMs be included in that trial? Do you think?
In the prior trials or the future trials?
The future one that's going to open.
I'm not sure of the exact criteria. It's probably still in dialogue. But in general, the idea is that you want the patients, let me comment on the registration trial that's running now. The intent is that the physician thinks they can remove 80% or more of the tumor.
Mm-hmm.
And so it's not required that it all goes away. In terms of location, it's generally believed that they need to be in the same surgical field. And that's because the tumors usually do touch if they're that close that they're in the same surgical field and felt to be probably the same tumor.
Would it make sense that once it's approved, you could use like IV to get these multifocal tumors?
Of course, that's exactly what we would like to do, and look forward to a time when we can explore IV.
Let me see. How far does the virus spread? Say you're injected in one spot. Do you know how far it could spread in what period of time?
Well, in general, it's interesting that the virus in various very scientific studies have been estimated to move about 1 centimeter a month. So these are modal cells. So if the virus that's infected moves, it will carry. If the tumor cell moves, the virus will move with it. But in addition, we do see virus that's in the, we put it in the tumor, but it ends up in the CSF or in the bloodstream. It's always been cleared on its own, but there is a chance for the virus to spread further than we put it in. And we have seen some examples on resections that there is a virus in tumors beyond the original one that was it was put into.
Good. Do you see a difference with the MGMT status of the tumors?
Yeah, in the recurrent setting, patients, which is the only place we've looked, we don't see a difference. It's small numbers, but so far we haven't seen it.
Good, very good.
Yes.
Does steroid use decrease the chance of it working?
We don't have strong data on that, but there is good literature that says it's better, if you have an immune therapy, it's better to avoid steroids. I would say that one exception to that is if you have brain swelling that the physician's worried about, they should probably give a course of steroids to control the swelling if it looks dangerous.
And this next question is going to be impossible to answer, but when do you think this will be approved?
Well, we have our interim analysis this month, and this quarter, and another one in the fourth quarter, or end of this year, I should say. And so hopefully one of those two time points, we've obviously would like to make this treatment available to patients with brain cancer as soon as possible.
Okay, hopefully you don't have to wait for that newly diagnosed trial to complete, like 4 or 5 years from now, right?
Yeah, exactly. That would. I'm glad we're running it because it's important for to explore this early stage treatment. But yeah, that would be a long time to wait.
Yeah. Okay. In the new trial, the control group will be allowed to use Optune, but not the treatment group. Do you think there's any interaction between Optune and your treatment?
We haven't really looked at it sufficiently to be able to respond to that, but there are theoretical reasons they could have an interaction. So we're really trying to compare ourselves to standard of care. So we've left it in the control arm because sometimes it's used, but we've felt that it's not used that frequently, that it's more balanced to not use it in our treatment.
Okay. This is a tough one. Is there any chance of the virus spreading to the loved ones?
We do have studies that look at that in mice. We look for mice that are living together, some infected and some not. We haven't seen spread in those studies. And so we're hopeful not. We evaluate saliva, urine, and blood constantly throughout the course of the studies. And in all patients, the virus clears. So they are cautioned to use contraception during the trial and for the first year. But so far, we're very optimistic that that's not going to be an issue. And I should point out that so far, everyone we've tested have antibodies to this virus. It's probably in our environment, and we've all been exposed, of course, at much lower doses. But it seems like people can control the virus.
Okay, and if worse came to worse, would the antivirals be effective against this?
Yeah, we've tested the antiretrovirals and have identified 3 that are highly effective against our particular virus. And that's, we educate the physicians so they're ready. But so far, we haven't needed to use these agents in any patient.
No, it's just nice to know they're possible.
Yes, absolutely. Not everyone behaves the same, so it's good to have a fail-safe.
Okay, that's about it for all the questions I had. Let me just check Facebook one more time. There's a couple more questions on compassionate use, so start working on that. But that's about it. I want to thank you so much for taking time on your Sunday evening to help us with this. And it's a very exciting project, and I'll be sure to tell everybody when the trial opens.
Okay, thank you, Al, for this and also for everything you do for us. We really appreciate it.
Thank you so much. Bye-bye.
Bye.